without further ado, I'm going to hand it over to Rob. Rob's going to give us the presentation on the OpenSPR and the complete test overview. So, take it away.
Awesome. Thanks, Daryl. So yeah, as Daryl mentioned, my name is Rob. I'm going to be giving the overview of today's webinar. So I really encourage you, this is a fairly informal setting, so use that Q&A button whenever the opportunity strikes. Daryl is technically my boss, but today he's working for the Q&A button. So he's going to kind of keep him on his toes while I'm talking. If there's anything that does involve kind of more in-depth discussion, I do have a section at the end where we can kind of have more of an open forum to investigate any questions that you have. So today's webinar itself, I've split it up into 5 main sections. We're going to start with the fundamentals. What is SPR? Why do we care about it? And then we're going to go more into the details of what considerations should I have when I'm designing my tests? How do I physically run my tests? And then once I've successfully run a test, what am I looking for in my data? Am I great? Am I getting a Nobel Prize? Or do I need to repeat my run? So by the end of today, you're going to be able to make that determination on your own. So we'll start with the basics. What is OpenSPR? So SPR is surface plasmon resonance, which is what SPR stands for. And there's kind of some terminology that we need to be familiar with before we kind of proceed with our discussion. So when you're doing SPR, you are trying to determine the kinetics of 2 binding partners by quantifying the change in mass at the surface of a sensor. So in order to do that, you have to immobilize one of your 2 binding partners onto that sensor itself. We term this binding partner the ligand. If you're from a biology background like I am, when I came into SPR and they were using the term ligand, I thought that is not what a ligand is. But please just put your biology aside for a second, and we're going to focus on SPR terminology. So the ligand is what is physically immobilized to the sensor. The other binding partner is termed the analyte, and that is passed across the ligand in a running buffer at various concentrations so that you can determine if there is specific binding between the 2 partners. So now that we know kind of the terminology, the next thing to understand is the actual steps involved in an injection during your SPR test. So there's 2 distinct phases that we're interested in quantifying. There's the association phase, which is typically represented by an increase in mass on the sensor as your analyte binds to your ligand at a higher rate than the dissociation is occurring. So you can see here on the figure, the association phase is usually associated with an increase in signal. And then there's the dissociation phase, which is where there's no new analyte being introduced into the system. So anything that dissociates off of the ligand is flushed across the sensor and into the waistline. It's not given an opportunity to bind again. These 2 phases are important. They're going to give us our K on and our K off, and these are what's actually used to calculate our affinity. So why are we interested in SPR? It's not just because your principal investigator told you were interested in it. The reason that SPR is great to use is because it has such a broad application. It's label free, so you don't have to worry about partnering one of your 2 binding partners to a fluorophore, a radioactive isotope, or anything like that. It will give you both the kinetics as well as your affinity. And the reason that it's nice to have kind of that more complete picture with SPR, I like to show this kind of figure on the slide you see here. If you look at the actual figure, you see a variety of different lines. Some of them look like there's a really quick association phase and a really quick dissociation. Others look a little bit different. Every single one of these lines has the same affinity. So K large D is 100 nanomolar. If you didn't know what your K on and K off were, you could look at these and say, well, they all have the same affinity. They all must be behaving in the exact same manner. Whereas when you have kinetics as well as affinity, you have a little bit more of a story of how do my actual binding partners interact to get me the affinity that I've calculated. So if you look here, there's a variety of K on and K offs that are presented for you on the slide. And those are what are actually mapped in the figure. And that's what gives you the same affinity. So having this is just kind of an extra piece of the puzzle when you're trying to determine what is binding, what isn't, and how is it actually interacting. The other nice thing is that the SPR is molecule agnostic. It works for a broad variety of applications, and it is becoming a critical step in research and discovery as well as drug discovery. You have to prove that the drug you're actually developing is doing what you say it's doing, and looking at and understanding the interactions and the affinity is a critical step for that. Another nice thing about SPR, you can use it with a variety of different sample types. So what do I mean by that? You can pass across something as a complex matrix, like a plasma sample, whole blood, and as long as you have a specific ligand that is only going to interact with one of the components of this complex matrix, then you're going to be able to derive kinetics. You can also use it for smaller molecules or small analytes, such as like an oligo, an aptamer, even DNA and RNA, or you can get into the larger analytes like the typical protein, cells, virus particles that a lot of people are interested in quantifying. How does it actually work? So there's two different modes of, or two different types, I should say, of SPR instruments. There's the traditional one and what we call the localized surface plasmon resonance, or LSPR. So Nicoya works with LSPR instruments, and you can see that is in kind of the blue box on the right-hand side of the slide. And so how this works is we use gold nanoparticles that are affixed to a sensor. And then you would then immobilize your ligand to these gold nanoparticles and pass your analyte across. The light source is directly behind the sensor. It shines through the sensor, past the ligand, through the analyte, and into an optical detector on the opposite side of the instrument. We're able to quantify the change in mass on the sensor surface by looking at the change in the absorbance wavelength. So you can see towards the bottom of that figure, you've got the figure what we're looking for. As your analyte binds, it shifts the overall absorbance, and that is what we actually use to derive a live signal when you're conducting an SPR test. If you're looking more towards the traditional SPR instruments, they use a single layer of gold across a sensor, and instead of looking at the change in wavelength, they're actually looking at the absorbance angle, the change in the absorbance angle. So the light source is positioned on one side of the sensor, it shines onto the sensor, and then it actually reflects back into an optical detector, which is on the same side. They're both going to give you accurate kinetics. The nice thing about the LSPR is that our systems are a little less susceptible to vibration and to temperature shifts because we're not worried about catching the actual change in an angle of absorption. So now that you kind of understand a little bit of the fundamentals, we're going to reflect back on the figure that I showed you on slide number 1. I know you didn't think there's going to be a callback so soon, but there is. So when you have your sensor, we're going to discuss sensor chemistries and stuff in a little bit. You have to do a couple of steps in order to prep it to make sure that you're going to detect your specific binding. The first one is to prepare your sensor. The activation of our sensors depends on what sensor chemistry you choose, but basically at this time, you're prepping the sensor to be able to immobilize a ligand. And that's kind of just demonstrated for you as this flat baseline here, where you're passing running buffer across the system, then you're going to activate your sensor. Once the sensor is activated, the next step is to do an immobilization of your ligand. And so this is kind of the dark blue in the center of the slide. So this increase here is showing you that you are immobilizing one of your two binding partners onto this sensor, and it's staying there. How do we know it's staying there? The baseline is not returning back to the baseline it was at before you started your ligand immobilization. If you have a baseline, you increase your ligand, it looks like, oh, great, I think it's there, but then all of a sudden it comes right back down or it decreases. That could indicate that you don't actually have ligand on your sensor. And we're going to talk about that in a little bit. So in this case, everything is going according to plan. We have our ligand immobilized, and then the next step is to test various concentrations of your analyte. So here you're going to see a response above the response baseline from your ligand immobilization. And depending on the affinity of the system, you may see a dissociation, which is pictured here, or it may be a high affinity system where you won't see as much dissociation. That's going to depend on the two binding partners that you're testing. So how do we get our kinetic constants from this kind of voodoo that we're doing on the sensor surface? So the first thing that I mentioned is we're going to look at kind of a figure as if we've already immobilized our ligand and we've now proceeded on to testing different concentrations of our analyte. So once our ligand is immobilized, running buffer is passed across the sensor until you inject the next or the first concentration of your analyte. So your first step, you're going to see a baseline that hovers around 0. And then you're going to have the association phase where the analyte that's in the running buffer that you just loaded into your sample loop is passed across the sensor, and it has the opportunity to interact with that ligand by diffusing out of the running buffer and binding with the ligand that's immobilized on the sensor. After that, there's a dissociation phase. So how our instruments operate during the dissociation phase, the active injection is over and the instrument switches back to just passing running buffer across the ligand. So any analyte that dissociates is swept across into the waistline, and you're going to see a decrease in the signal as the mass of the analyte leaves the ligand and is removed from the system. Sometimes there is a need to regenerate. So this is what kind of this large box-like shift looks like here. We're going to talk about regeneration in a couple of slides, but the important part to understand here is that regeneration may be necessary if you have a high affinity system and you want to make sure that your ligand is equally available to bind every concentration of analyte that you're going to try. So the final kind of, let's wrap it up in a nice little bow, slide for what is SPR is just to associate ourselves with some more definitions. So if you see Ka, K lowercase k, lowercase a, that can also be called K on or your association rate constant. If you see lowercase k, lowercase d, that's referred to as the dissociation rate constant. It can also be called K off. And then finally, if you see uppercase K, uppercase D, that is your equilibrium dissociation constant, and that is what's used to calculate your affinity. So those are kind of the three major terms that you're going to see when you're running your SPR tests. And if you're familiar with what each of them are, you can assign them to where each is actually occurring when you're running analyte injections. Okay, so now you're an expert and you're going to be testing or running the next webinar. But before we get to that, we are going to go with how to plan an experiment. So you know the fundamentals of SPR, you know your two binding partners that you're interested in working with, but how do you know what to do with them? And that's kind of what this section is going to look at. So the first thing that I like to consider is which one of my two binding partners am I going to immobilize on the sensor surface? There's a couple of factors that may make one better than the other. The first one is size. So we generally recommend, if possible, immobilizing the smaller of your two partners on the sensor surface. The reason we say that is because if you have a larger partner as your analyte, which is going to be passing across and then binding to your sensor surface, that's going to induce a larger response in your profiles, and that will be should make detection a little bit easier. That's not always possible. Sometimes the smaller of the two is more applicable to be your analyte, so you don't have to just consider size. Other things to think of are the purity. Is one more pure than the other? We would recommend purity is very important, and you would want a pure ligand on your sensor surface. The number of binding sites. Whenever you're running a test, you're trying to choose a fitting model. We like to try and minimize the number of binding sites that a ligand has. So if you have two partners, one will only bind the second one once, then that would make it more kind of optimal to try and make that one your ligand. Charge, depending on your sensor chemistry, you may want to try and immobilize opposite charges, which will increase the amount of immobilization that you can achieve. Once you've kind of considered those four factors, the next thing to think of is, well, how am I actually going to get my ligand on the sensor? And sometimes you'll have tags that can actually work in your favor. So for instance, maybe one of your two binding partners has a his tag, it has an FC region, it's biotinylated. Any of those, you can actually select a sensor chemistry that will reflect that and will work with you to immobilize that specific partner. If you don't have something like that, you can always default to a carboxyl sensor, which is basically a covalent binding between whatever free aiming group you have within your ligand and the sensor surface itself. So now you've kind of identified which one I'm going to make the ligand. How much do I now need to put there? So there's no kind of one magic number fits all, but what we like to do is use this kind of equation you can see at the bottom, where we will look at the molecular weight of your analyte, the molecular weight of your ligand, and calculate how much a ligand immobilization do you need to see in order to successfully move on and have the best chance of seeing your analyte interactions. So to do that, we have R max. So RU is our response unit. That's what's reported in the OpenSPR software. And you will use this. We recommend setting R max to 50 at the minimum. The reason we say that is because 50 is well above the background noise of the instrument. So you know if you're seeing a minimum of 50 response units, then you are going to have a good chance of seeing your specific binding. So by setting Rmax to 50, knowing what your analyte and ligand molecular weights are, you can then solve for what ligand immobilization response you should see when you're doing a ligand injection, and it will tell you, am I actually getting enough on the sensor surface? So I mentioned in the past, or in the previous slide, that we do have a variety of different sensors that you can order depending on what your binding partners are looking like, whether they're tagged, etc. So the one thing to consider about this is when you're looking at what sensor chemistry may work best is to determine what kind of immobilization you're actually going to achieve. So what I mean by that is sometimes if you're going with a covalent coupling, then the ligand is irreversibly bound to the sensor. You're not going to be able to regenerate the system by removing the ligand itself. So anything that you immobilize there, it's going to stay there forever and ever, amen. Whereas when you're looking at some of our capture coupling sensors, you do have the option to potentially strip both the ligand and the analyte off. For instance, if you're using our NTA sensor, that is a his tag, and we do have imidazole, which will outcompete whatever is his tagged onto the sensor, and it allows you basically to flush everything off, and then you can re-immobilize your ligand and start with another concentration of your analyte. So these are all kind of considerations. The nice thing about this is you don't have to memorize it. On our support portal, we have some great technical notes that go through the intricacies of each of these sensor chemistries. So if you're interested in that, you can go on our support portal, you would navigate to whatever sensor you're going to work with, and you can read a little bit about what steps does it take to activate it and what considerations should I have when I'm doing my immobilizations. The next thing that I'm going to try and really drive home, if there's only one thing you take from this, it hopefully will be this key fact. When you're running your test as a new user, I cannot stress using the ligand wizard enough. So the ligand wizard is just an option you choose in our OpenSPR software. And basically what it does all the hard work for you. It's going to ask you a couple of questions, such as you can see in the top capture picture here. It's going to say, what sensor are you using? So you would just select whatever sensor you did and click start. It's then going to ask you, what's the molecular weight of your analyte and your ligand? And it will do the calculation for you about how much response should you see when you immobilize your ligand. The other nice thing about the ligand wizard is it will automatically control flow rates and flow patterns. So you don't have to remember, okay, I need to change the flow rate to be slower or faster, etc. It's all done for you if you select the ligand wizard. Once you've run the ligand wizard, so it will walk you through the steps to prep whatever sensor you're using, the ligand immobilization itself, and then it will do a calculation for you. It will look at the baseline before and after ligand immobilization, and it will do a calculation and give you a pop-up similar to what you see at the bottom of this slide, where you can see the immobilization level in this example was 383 response units. We needed a minimum of 13 response units. Did we reach our target? Yes. The nice thing about this is if you're unsure of what ligand concentration you should have started with, maybe you put one in that wasn't high enough and you don't reach your minimum threshold, then you have the option to inject additional ligand before proceeding on to the rest of your test. So your sensor is not a loss. You go back, you inject a little bit more ligand, and the wizard will look at what is the change in the baseline after the second injection as well. So it's very nice. I really recommend doing this. One of the most common mistakes, I should say, that new users make is when they're not using the ligand wizard, they're not getting enough ligand onto the sensor, they're proceeding on to test their analytes, and there's simply not enough ligand there to visualize your binding. So you're wasting your analytes, your reagents, and you also run the risk of potentially contaminating your reference channel. So what do I mean by that? In a two-channel system, we have our reference channel, which is also called channel 1, and our active channel, which is called channel 2. We only immobilize ligand in channel 2, and the ligand wizard makes sure that that injection pattern is followed. If you don't use it, then you have to use your own brain power to remember, oh, I make sure to change the ligand or the flow pattern myself before I start my ligand immobilization. So now we're going to pretend we have a great ligand concentration on our sensor surface. It's optimized. Now we're going to proceed on to our analyte concentrations themselves. So how do we know what analyte concentrations to start with? There's a couple of factors to consider. Number one, is there any previous information, either within your own lab, within the literature itself, which tells you the suspected affinity between the two binding partners that you're testing? If you have that number, we would recommend testing an analyte range concentration from 0.1 times up to 10 times that KD. This is going to hopefully generate a kind of response that you see at the bottom left of this slide, where each injection of your analyte concentrations, you're going to have a concentration-dependent increase in response. And it's showing you that you're not oversaturating or undersaturating the ligand itself because you have a broad response across all of your analytes tested. If you don't have a suspected KD, which is the case in a lot of research, then what we would recommend doing is starting at the low nanomolar concentration and working your way up until you start to see responses that are similar to the bottom left-hand side of this slide. If you're running an analyte injection series and you notice that your analyte injection responses seem to be all centered towards either the bottom or the top of your space, then this can indicate that you're either testing an analyte concentration that's too low or too high. So if you notice response profiles like this, you don't have kind of that nice concentration-dependent response, we would recommend analyzing what range you're testing and making adjustments accordingly. Once you have your kind of analyte range determined, we then recommend running your samples or running your analytes at least 3 to 5 different concentrations. More is better if you can do it, but it's not always required. What we don't want you to do is to do a single injection of a single concentration of analyte, calculate your kinetics from that, and say, I'm the best and no one is allowed to question me. So what we like to do is say, try at least 3 to 5, and then see how those range and base your kinetics off of that. So the next thing to consider, step number 4 as you're running your test, is your regeneration. So I touched on this a little earlier. Is regeneration always needed? The answer is not always. So it all depends on the affinity between your two binding partners. For instance, if you have a lower affinity system, you may have a fast off rate, which is the red writing towards the bottom left of the slide, where you can see the association phase ends with the dashed line. The dashed line is where dissociation starts, and it's a very quick drop off as the analyte dissociates from the ligand. If you have something like this, you can wait until you have natural dissociation. 100% natural dissociation might take 5 minutes or so, which is not that long to wait between injections. However, if you have a high affinity system, such as the right-hand side of this slide, where after the active injection ends, you don't see much dissociation at all, it doesn't make sense to sit and wait for 10 hours between injections until your analyte naturally dissociates off. If you have a slow off rate, this is where we would suggest initiating a regeneration injection in order to kind of speed up the time that you have to wait. So in that case, a regeneration, you would standardize your length of dissociation, be it 5 minutes, 10 minutes, etc. we would hope that you would standardize this to capture 5% natural dissociation. As long as you have about 5% dissociation, you can regenerate at any point after that. So let's say you're going to wait 5 minutes, and then after 5 minutes, you will inject your regeneration solution. This should return your system back to the baseline it was at before you injected your analyte itself. How do we choose which regeneration solution to use? There's no kind of one size fits all. We have a really nice chart on our support portal that we have shown with typical applications like a protein, antibody, antigen, etc. And we have some suggestions as to what we would use. But what we would recommend running is either a regeneration scout to see what solution is going to be best, or you may get lucky and find in the literature someone has published something that they have used in the past. You could start there and determine whether or not it's actually working for your system. How do we know if it's working? Great question. So what we can see here, there's 3 different traces on the bottom of our slide. And there's the 3 different answers as to, is my regeneration working? A good regeneration is going to take the baseline after your analyte injection, reset it back down to where the baseline was before this analyte was injected. So you can see here, this is under the good regeneration figure. If you choose a regeneration that's too weak, then you'll see a decrease in the baseline, but it won't go all the way back down to where it was prior to your first analyte injection. This is telling us that we removed some of our analyte, but we weren't successful in removing all of it. So any new injection of analyte, there's not the same amount of ligand on the surface that's capable of binding, and we may see different responses because we didn't actually strip all of the analyte from our previous injection. The third option is if you went too harsh, you chose something that's just going to go in there and basically completely disrupt everything, then what's going to happen here is your baseline is going to drop below the area that it was at before your first analyte injection. What does this mean? It could mean a number of things. It could mean that you perhaps removed a little bit of ligand from the sensor surface itself. It could mean that you accidentally altered the configuration of the ligand, so it's no longer going to be capable of responding to the analyte as you subsequently regenerate. But overall, we're looking for the good regeneration where the baseline is in line with where it was before the analyte concentration injection. To run a regeneration scout, what we typically recommend to do is you immobilize your ligand, you select one concentration of your analyte, a higher concentration that you know elicits a response, and then you would inject that across your ligand, try regeneration solution number 1, and then you'd inject the same concentration of your analyte across the ligand again to measure how do my responses actually compare. If you see that perhaps the regeneration is too harsh and it damaged the ligand, there may be no response during your second injection. So that's why we recommend sticking with a single analyte concentration. It allows you to compare apples to apples when you're looking for your actual, what regeneration would work. If you have multiple regeneration solutions that do work well for you, what we recommend to do is to default to the least harsh option. That way, you're not treating your ligand carelessly. You're trying to give it as much TLC as possible. So you would choose the least harsh regeneration solution that still gives you good regeneration. Okay. Sometimes, though, I'm not going to lie to you, you're not going to find a regeneration solution that agrees with your ligand. In that case, there's a couple of options that you have. Number 1 would be not to run a regeneration throughout the course of your test. Or number 2 would be to flip the system. So flipping the system would be to take what you currently have as the ligand and making it the analyte, and taking what you currently have as the analyte, making it the ligand. This is a really nice option if you notice that your ligand seems to be unstable after a regeneration, or if you're having poor specific binding, then perhaps the actual binding site is a little bit, it's not available. I just had completely lost my train of thought. It's not available when it's actually immobilized onto the sensor, Or another thing, if you have high non-specific binding, then what you could see here is by flipping the system, you may actually be able to decrease that non-specific binding. It is not a one-size-fits-all. Sometimes you have limited utility if you can't immobilize one of your two partners, or if one of your partners is much, much smaller than the other, and if you were to use it as an analyte, it may be a little bit difficult to detect the binding just because of the size of the analyte itself. Okay, so now we're going to take a breath. I'm dumping a lot of information on you, and I promise this will be available to you as a recording afterwards. But we're going to say we have identified who we're using as our ligand, who we're using as our analyte. Do we need regeneration? We have everything ready to go. We've selected what sensor chemistry we're actually going to be using. We're going to physically run our test now. So there's two different options for your test. The first is a manual OpenSPR hardware, which is what you see on the screen here. So if you have a manual system, you are going to interact with the instrument at the injection port. So this is kind of that blue circle you see on the upper right-hand side. Well, it's on the upper left-hand side of the instrument when you're facing it. This is where you're going to load everything that's going to pass across the sensor. There's a sample loop in there, so you can load a sample a couple of seconds before you're actually injecting it. One thing to remember is when you load a sample, it's not actually connected to the rest of the instrument until you click start or until you click inject on the software. So if you load something into the sample loop, turn around and look at your laptop and nothing happens, you haven't actually introduced it across the sensor yet. So once you've loaded this, if you lift the lid of your instrument, which is kind of this OpenSPR, you are going to see what is pictured on the right-hand side of this slide. So what I was telling you about before, we have light sources here. The light source in this picture is kind of that red, almost looks like a USB key being plugged in. So this is shining through our flow cell, which the flow cell is a two-piece section within your instrument. There's the hard outer side, and then there's the soft, smaller inner side. The soft, smaller portion is what actually comes into contact with your sensor. It's where the actual fluidics channels are carved and where all of the contact between your sensor and whatever you're injecting occurs. So this is the flow cell itself. And then if you kind of work your way more towards the right, we have our sensor stage, which is where the sensor is actually loaded into the system itself. In this picture, the sensor is undocked from the flow cell, so it's not in contact. Whenever you're running a test, you would have the lid closed and you would have the sensor docked onto the flow cell so that there's a physical touching between the two, and you're going to have interaction between whatever you're passing across the sensor and the sensor itself. The final kind of thing to look at here is along the to the far right. This is the optical detector. So there's two fiber optic cables in there. They're trained on channel 1 and channel 2 respectively, and they will give you live data reports as you conduct your tests. So this is really nice. You can quickly see how is my reference channel acting in response to my active channel, and it kind of gives you a way to quickly orient whether or not you want to continue, you want to increase or decrease concentrations, etc. So the flow cell, these figures you see on the bottom here, this is as if you're looking head on to this flow cell itself. There's two different flow patterns that you can select. So the first flow pattern is what we call both channels, or channel 1 and channel 2 path. And what would happen is your liquid would come from the lower left-hand side of the flow cell. It would flow up channel 1, across the linker, and then down channel 2 into the waistline. The other option for flow that you have is channel 2 only. So instead of coming from the bottom left, the liquid is going to come from the top of the flow cell. It's going to flow directly down channel 2 and out into the waistline. Channel 2 only injections are typically used for ligand immobilization only. Everything else is going to go across both channels, even your analyte injections. And the reason that we suggest this is it gives you an immediate check and balance system to see, is there any interaction or any responses that I'm seeing in channel 1 when I inject my analyte? If there is, I know it can't be specific binding. I don't have any ligand there. So it must be non-specific, and I must be able to try and mitigate it by using a variety of different factors we're going to talk about in a couple of slides. There we go. I was going to say it froze on me. So the next thing to look at on your manual instrument is the far right-hand side. There's 4 different bottles. So this is buffer bottle 1, 2, your DI water, and your waste. The 2 buffer bottles are interchangeable. So if you needed to use 2 different running buffers over the course of your test, you have the option to load 2 different ones here. If you're just running the same running buffer throughout the whole course, you can just use one of these running buffer bottles. So it does give you some flexibility. Why would you consider using 2 different buffers? If you have components of a buffer that will interfere with a ligand immobilization, then we would recommend making up buffer bottle number 1 without those components, buffer number 2 with those components. And then you would switch between them after your ligand immobilization. The DI water bottle, you can keep water in here. We recommend usually about 200 mils or so. It can help with cleaning. It can help with putting the instrument in standby using water. So it's nice just to keep some water in this when you're running your test. And then the 4th bottle, which would be the furthest away from you if you're standing at the bench, is the waste lines. So there's 3 waste lines that come into this. And basically, they come from 3 different sources within the instrument itself. One comes from what passes across the sensor, one comes from any overflow from your sample loop, and one comes from the pumps itself. So everything, all liquid waste generated is going to end up in this waste bottle. One thing I really like to do, or I should say I really encourage you to do, before you start your test, take a glance on the right-hand side and make sure that waste bottle is not going to overflow throughout the course of your test. It's not a high volume system, so even if you have maybe 10, 20 mils in your waste bottle already, you're totally fine. But it's a pain in the butt to come in and say, oh, I've made a mess. I should clean this up before my boss finds out. So if you just make sure that the waste is empty before you start, you're going to be good to go. So now we've checked how does our instrument work? Is the light source actually working? We have our waste bottle empty. Now we need to put in our sensor itself. One of the great features about the OpenSPR software is that the pop-ups give you step-by-step direction on what you should be doing when you get to a specific part of your test. So for instance, if you're loading a sensor, you would click on, okay, I'm going to load a sensor, and you'd see this pop-up, which is on the screen for you. It walks you through exactly what to do. Our sensors are sent in little vials with these cute pink caps, and basically there's up to 10 of them in there. They are shipped in a buffer, so you don't want to uncap it, turn it upside down, and then you'll lose your 9 sensors and all of the buffer all over the bench. So what you would do is you would just take the sensor out of the vial. The main goal of the entire sensor prep is just not to scratch where the gold nanoparticles are on the sensor itself. Everything else is glass, so we wear gloves, use tweezers, and as long as you're not dragging your tweezers across where that gold nanoparticle section is, then you can prep the sensor according to the pop-ups. The other thing to note is the orientation of your sensor. If you try and put it into the sensor holder and it's sticking up too far, then you're just going to want to make sure that the cut is in the lower right-hand side of the sensor itself. This is the way to orient, make sure the gold nanoparticles are facing the flow cell, and that it will lay actually flat or more flush into the sensor holder itself. So the next thing to consider, you've loaded your sensor, you've got everything ready to go. Before we get into what a test would look like when you run, there is a question that I have. I'm just going to put it up as a really quick poll. We have an auto sampler option. So what you can see here on the right-hand side of this picture, there is the manual instrument. On the left-hand side, we call this the auto sampler, or sometimes we call it the XT. If you have an XT, everything that I just described to you holds true, except for the fact that you're not doing any physical injections through the injection port itself. Instead, all of your samples are loaded into the XT, you build a test timeline, and then you walk away and you come back a couple of hours later when your test is finished. So I see here, there's only about 10% of us have XTs at the moment. So we're still going to go through this information. But if the 90% of you don't have an XT, I will share the fact that we can modify a manual instrument to include an XT. So if this is something that is interesting to you or to your team, you can always contact us and we can walk you through how this would occur. So the XT itself, basically, if we open up the lid to this, you can see inside. So there's two different bottles that you're going to interact with. One is on the right-hand side. You want to make sure this is filled with water. The other one is inside the instrument itself, and you want to make sure this is filled with 80% isopropanol. These two components are used to clean the sample loop after each injection. So if they were to run dry throughout the course of an experiment, you have the potential to introduce air into the system, which is something we want to avoid. So as long as these two are topped up before you start your test, you're good to go. The other thing to look at is we have a cooling cover. So you have an option to set a temperature to hold your samples at before they're passed across the sensors. And you would just slip the cooling cover over top in order to help maintain that temperature a little bit more steadily. If we remove the cooling cover, you'll see that there's a tray here. You can put two 96 well plates there. They're in a left and right orientation. When you do load them, A1 is always going to be closest to you. So just so you can easily orient how you're putting in the plates. And then what happens is you tell the software what samples you put in what wells, and the needle will move along this robotic arm to wherever the sample is. It will descend into that well, draw it up into the syringe on the side, and then actually pass it across the sensor in the steps that you are indicating you'd like to test first. The nice thing about this, if you have the cooling cover, it is designed not to interfere with the robotic arm and the needle itself. So I don't want you to think, well, I can't use it because then the needle will all of a sudden break or you'll have some interference. As long as you're not shoving that cooling cover in as hard as you can, it does have a natural stop and it won't interfere with the needle. So if you are using the XT, the auto sampler, then there's a couple of extra steps to do before moving on to the actual manual or the actual injections. If you're using a manual injection, then these two screens are not going to be seen when you're setting up a test. So the manual, we have some really great, it's called the Pro Tips series. It's on YouTube, and it goes over how to do physical injections with the manual instrument. I'd recommend you can go on YouTube, look up Nicoya, and those 6 videos will start. I'll save you a little bit of time. It's video number 3. So if you don't want to watch kind of the whole setup and everything, you can go right to video number 3. But for the XT itself, this is the screen that you're going to see first. So basically, you see the two 96 well plates, and you will indicate on there where you've put what samples. It can be in any order. It doesn't have to be in a specific fashion. Just make sure you know where it is that you've put each individual sample. You click on it, and then you add the name as well as the concentration of the sample. If you're doing multiple wells of the same sample, you can click on multiple wells at the same time and add them all together. Once you've loaded them all or logged them all, they'll appear here. They'll also appear grayed out, so you can't accidentally assign multiple sample types to a single well. The next thing to look at is on the right-hand side. So this is building a test timeline. In your XT, this is exactly what you're going to see. You have a bunch of different injection processes that you can choose from. You just drag and drop them under the test timeline in whatever order you like. The ligand immobilization feature is this purple key right here. So if you were to drag and drop that over, you'd see something very similar to this, where it's asking you, what would you like to do for surface cleaning, for surface prep, and to actually immobilize your ligand. In the XT, your samples are shown for you on the right-hand side until they are assigned to a specific well. Once they're assigned to a specific injection, they disappear from here and they show up there. If you're using a manual instrument, then once you have started your test timeline, which is just by clicking this button here, you're going to give a name to your test, which is automatically saved as the date and time you've run it, and then the name that you're assigning to the test. And you're going to see the ligand wizard be a button kind of between the save and delete in this screenshot. So you would click on that to start your test, and it would ask you your questions about what is your molecular weight for analyte and ligand, and what sensor type are you actually using. So once you've either built your test timeline with your XT, or you've started the test with your manual instrument, there we go, you're going to see your baseline, your live report of your baseline. So what we have here, I have just taken a video for, let's see, just have to drag that up. Oh, the video is not going to work for me right now. Bear with me. See, I knew technology was not going to let me escape unscathed today. But basically, what we're going to look at when I get this working, okay. So what we're looking at here is what you're going to see during an actual test. So you can see here, this is the XT that I set up. On the left-hand side, you see the ligand immobilization. It walks you through what each individual step is. And when the step is completed, it grays it out. So you can kind of see very quickly where you are in your test. If you're using the manual, then the ligand immobilization would work in a stepwise fashion. It would say, please inject the surface cleaning, please inject the surface prep, inject the ligand itself, and you would click next for each to proceed through each of them. But what we like to look at is along the top. So this kind of software tools that you can use, you can set the temperature. So because it's an XT, there's two different temperatures here. The first is to set the temperature of the 96 well plates that are holding your samples. And the second is to set the temperature under the actual sensor / flow cell itself, which is what the interaction is going to take place at. The next thing to look at is it will tell you what your running buffer is. So we are currently running our buffer out of bottle number 1. You do have the option to rename these. So for instance, if this was PBST, we could have named this PBST. So I recommend if you are switching back and forth between bottles, it's great to name this just to kind of quickly orient yourself. The next thing to look at is the flow rate. So the flow rate here, right now it's grayed out because in active injection, you can't change the flow rate. It's at 20 microliters per minute. A manual injection will allow you to change this after each one. In a time or in an XT, you've already set the flow rates when you build your test timeline. The next thing to look at is the flow pattern. So right now, because we are immobilizing our ligand, which is a biotinylated oligo, we're just looking to immobilize in channel number 1. Sorry, channel number 2. Don't quote that part. So channel number 2 is represented by the blue line we see on the data, on the live data feed. And you can see that we had a really nice response. It went up to just about 600 RUs of immobilization. And the red line, which is representing channel number 1, is flat. So there was no change in mass in channel number 1. We didn't immobilize any ligand there. So this is really nice. It will tell you very quickly how well is the ligand actually being immobilized, what is my RU. I'm going to fast forward it just a little bit because I'm not going to force you to sit and watch all 9 minutes of this injection. But basically, if we fast forward, we can see now that the injection has ended. How do we know that? This dashed line is showing us that the active injection is over. And so we're seeing a little bit of a decrease in our ligand, but it's not coming all the way back down to the baseline. So what we're hoping to see is that it stabilizes somewhere above where the baseline was prior to the ligand immobilization. If it does, the ligand wizard will then pop up. It will give you the overall ligand immobilized, the response units, and it will ask whether or not you would like to proceed. So here, what we're looking at, the solid black line indicates the start of an active injection. The dashed is the end of your active injection. And then you just want to make sure that you give yourself enough time to quantify, is the baseline steady at a certain RU after I immobilized my ligand? So even I'm going to increase it just a little bit more to about 8 minutes or so. And we'll be able to see that it's, so this is live. Well, it's a video, but it's pretend it's live, and the baseline has stabilized after a little bit of loss at the very beginning. So from this, we would consider this a successful immobilization. If you were really unsure, then you could increase your dissociation time as long as you like, just to make sure that it is staying there. But in this case, we were using a demo test that we have optimized within our lab here. So we know that it's going to be pretty stable at about the 575 RU. So the next thing, we've immobilized our ligand. Our next step is to go on to the actual analyte injection itself. So what I recommend doing, whether you're doing a XT or a manual, after you have immobilized your ligand, I like to zero the software. So you would just click on this zero option at the top. What that does, it removes any injections that you have displayed on the screen, doesn't delete them. You still have them when you go through your go through your data itself on TraceDrawer, but it removes them from visualization. And why I say that is because when you're starting with low concentrations of analyte, sometimes those responses are going to be much lower than what you achieve during ligand immobilization. So if you have 600, 700 RUs of ligand immobilization and your first lowest analyte concentration only gives you a response of maybe 30 or 40, that can be very hard to visualize on a scale that includes your ligand immobilization. So by clicking 0, you're going to see a similar baseline to what we're seeing now. So both channel 1 and channel 2 are just going to kind of oscillate back and forth for a little bit. You're going to then start your analyte injections. So when you're running a test, we recommend starting from your lowest analyte concentration and working your way up. Some people will do it opposite, but then you run the risk of either oversaturating or saturating your ligand and not seeing any responses, or if your regeneration isn't great, then you have already used up much of the ligand that's on the surface. So starting low, working your way up is the way to go. And basically what we're going to look at here, so once again, we've got our black line, which is telling us where the injection is starting. The other kind of features that are fairly nice to look at, if you look at the top right-hand side of the software, it gives you the current time remaining in your injection. So in this case, it's about 8 minutes and 20 seconds. And then it will give you the total test time remaining. So about 43 minutes here. If you have a manual, what this will say instead is time since last injection. And what this can be used is to standardize your dissociation time. So for instance, if you're running a injection, you know, all of your flow rates are going to be 20 microliters per minute, and you want to have an 8-minute dissociation time, then you would just wait until the time since last injection displayed 8 minutes, and that's when you would begin your next analyte, or either your regeneration or your next analyte injection. So if you're using the manual instrument, I don't want you to think you're going to see the current time remaining. Instead, it would say time since last injection. So now we'll just kind of draw our attention back to what's happening on the live feed. So we're only seeing a response in the blue line as we pass our analyte across, which is great. It means that we're not seeing any response in the no ligand containing channel number 1, which is the red line. If we did see a response there, it's probably due to experimental artifact, non-specific binding, something kind of odd is occurring that we would want to address in subsequent injections. But for us, in this example, we have a nice response profile. It's going up, it's going to go up even higher than 800 RUs at the moment. I'm going to fast forward it just a little bit so that, once again, we're not kind of waiting for the whole thing. And you can see it does start to reach an equilibrium phase, and then the injection ends, as indicated by the dashed line. So this is where we're now going to determine, do I need regeneration? In a system like this, we were using 2 complementary oligos. They have a very high affinity for each other, so we're not expecting to see too much dissociation over a very short period of time. And this is indicated right now. You can see the baseline isn't really decreasing after the injection has ended. So whatever has bound to the ligand is staying bound there. On the flip side, if you had a lower affinity system, you may begin to see dissociation, see that blue line start going right back down towards the red line as the injection ends. If that were the case, you may not need your regeneration. But for us, in this specific system, we know that we do. So the other thing, I'm just going to fast forward it even just a little bit more. And we can see, so even almost 8 minutes out, the dissociation is very minimal for this system. All right. So now what we're going to discuss is, in our heads, we have run 5 different concentrations of analytes, similar to the video that we just saw. We end our test on the OpenSPR software, and you have the data file created once you've ended the test. If you try and do any analysis while the test is ongoing, you don't have any data to work with, don't panic. It's not generated until you actually end the test. When you do end the test, there's a couple of options. If you're planning to reuse that same sensor, maybe you have a regeneration that will allow you to test a different type of analyte, then you can start a new test using that same sensor. One thing that we recommend doing is, if you're going to reuse a sensor, try to keep it docked in the instrument. Do not undock it, put it in the fridge, 2 days later, come back, try and dock it again, prime the instrument and use it. If you're going to be reusing a sensor, it's preferred to keep it docked and to leave the system in standby using the running buffer that would keep that ligand happy. Depending on what it is, it's going to vary. We recommend, if you're going to leave the instrument in standby for an extended period of time, switch to water, and that helps to flush out the system. It also ensures that if standby is interrupted, you're not going to have any clogging occur within the fluidic lines themselves. But if you're going to, maybe you finish your test Monday morning or Monday evening, you come back Tuesday morning, you're going to be using that same test, same sensor, then you would just put the system in standby using the buffer that you were using the day before. Okay, so that was a little bit of a tangent. But what we're going to discuss now is the data itself. So there's a couple of ways to determine what kind of data am I generating. The first one, this is the feed, or the live feed we would see during our OpenSPR. So you can see here, both the red and the blue lines are reacting in a very similar manner. If you are injecting your analyte and the red line is increasing to the same degree, sometimes even higher than your blue line, you have very either poor binding, or there's non-specific binding that could be kind of obscuring your specific interactions. This is kind of visualized exactly how you would see on the left-hand side of this screen, where the red line is going to react in a very similar response amplitude compared to channel 2's line. If you were to switch to the corrected view in OpenSPR, it would look fairly like a flat line, because you're basically subtracting out the same signal that you're seeing in your actives channel. If this is happening, there's a couple of options. So the first one that we would try and investigate is non-specific binding occurring. So if the signal is large, it means that you're not seeing no binding. You're probably having your analyte interact with your sensor in a non-specific way. So you're getting a false positive, basically. Non-specific binding can be caused by a number of different factors. It's not always going to be this exact one, so fix that and you're good to go. But what we need to understand is that it's basically an interaction between the sensor itself and the analyte. It's not due to binding between the ligand and the analyte itself. So if you are kind of trying to identify non-specific binding, maybe you ran your whole test, you created your data set, and then you import it into TraceDrawer, which we're going to talk about a little bit later on. Then what you're going to see here is signals that look very similar in both channel 1 and channel 2. So the corrected signal is this kind of light green turquoisey thing along the bottom, whereas channel 1 is red and channel 2 is blue, and they are seeing very similar signals. If you see non-specific binding, there's a variety of different ways that you can mitigate it. We have a great kind of support article on our blog, and it goes through multiple different ways to reduce non-specific binding. So if you are running a test, you see you have non-specific binding, you can check out this blog, and it will give you some ideas of components to add to a running buffer. There's a whole bunch of different ideas, and you can try 1 or 2 of them, and hopefully that will help you mitigate the non-specific binding that you're seeing. Another artifact that you may encounter is mass transport. So mass transport can occur if you have so much ligand on the sensor surface that it's not actually giving you an accurate K on, association rate. Instead, the high concentration of ligand on the surface is grabbing absolutely any analyte that passes it by. This is visualized by a linear response during your association time. So you can see here during the association phase, we have an almost linear response because we have put too much ligand on the surface. And instead of getting an accurate association rate, instead we're just seeing the Km. So it's a diffusion coefficient, and we're actually measuring that due to the increase in ligand concentration. If you notice afterwards, you're processing your data and almost all of your binding looks linear, similar to the red overlays that you can see here, there are a couple of ways to compensate for it. Once again, I'm going to refer you to our blog, but basically you have a couple of options such as increasing your flow rate, choosing a different fitting model if you've already finished the test, so all is not lost. But the first thing you have to know is how to identify it in order to rectify it. So if you're noticing that, then I encourage you to check out the article for the ways to limit mass transport. And then the final one that I wanted to talk about, it's a very common one that we can see, is called bulk shift. Bulk shift can also be called the DMSO effect. And basically what it is, it's characterized as a sharp response that is seen when you start an injection. The response can be positive or negative. So in the first figure you see here, both the blue and the red lines are examples of bulk shift. And it's caused when the running buffer you're passing through the system isn't matched to the buffer you've used for your analytes. So sometimes we can see this when even the running buffer, even if it's the exact same thing, the running buffer has been in the system for a couple of days versus your analyte buffer is freshly made. So it doesn't always have to mean it's equal in components or concentrations of various components. It can mean something as simple as age. So what we like to do is recommend that you try and buffer match as much as possible between the running buffer and the buffer that you're using when you're injecting with your analytes itself. And then, spoiler alert, I'm sure you know what I'm going to say, but we do have a nice article here as well that gives you kind of an idea for how to deal with bulk shifts. Is all data lost if you have a bulk shift? Absolutely not. So I don't want you to think, oh, Rob's lying to me. He said I could deal with this. I promise you can, especially if you're using a 2-channel instrument. A bulk shift can be automatically subtracted out, but you do have to know if one's occurring in order to try and mitigate it. Some other things that I just thought I would show you very quickly. If you're running a test and you notice some erratic responses, whether those are like really quick air bubbles that are passing through, whether it's a drift in the baseline, either up or down, what these can indicate is that there's some potential residue from previous tests in the fluidic lines that are causing problems with your current test. So if you notice a really drifty baseline or really erratic responses, what I would recommend doing is cleaning the instrument. In general, we recommend running an instrument clean, a basic clean, after every 5 or 6 times that you've used the instrument, and then running an advanced clean about once a month. Depending on the usage of the instrument and the type of injections or the type of samples that you're using, you may need to increase the frequency of these cleans just to make sure that the buffer remains, or sorry, the response profile remains nice and uniform. All right, so we're almost there, I promise you. This is our last section, and I thought instead of forcing you to watch me talk about slides, I would actually show you how we process data using TraceDrawer. So to do that, we're going to go out here. And basically what we recommend is, so you've finished your test, you've created your data file, and then you're going to open up TraceDrawer. So this is included in the laptops that Nicoya sends out. You should have a license key that is already on that laptop, so for you to put in if you haven't actually started the TraceDrawer. So once you open it up, you're going to see a screen identical to this. In order to add any data, you click on Add Run down here on the bottom, and it's going to open up wherever it was that you last were. So our data is actually stored under Documents, OpenSPR, and then Test Results. When you open up that, any test that's been run on that laptop is displayed for you with the date, time, and name. So we ran a test recently that we're going to analyze. So what you would do is you would just select whatever file you're interested in working on, and then you're going to see that there's 3 different folders. We're interested in looking at the trace drawer export. The other 2 are more instrument log type things. So if you're interested to see what time did the valve change from channel 2 only to channel 1 and 2, or what was my response at a specific time, you can click on these and you can look at the data itself. But for the kinetics, what we are going to do is we're just going to click on the trace drawer export, and you're then given 3 different options. So these are just different resolution of the same data. It's just different number of data points that are presented per second. We always work with the normal resolution. These have been used in publications. They're really nice to process through trace drawer. You're welcome to use either of the other 2 if you prefer, but for today, we're going to start with our normal resolution. So by clicking on that, you once again have 3 different options. So there's option for channel number 1, channel number 2, and your corrected data. I like to import all 3. You're only going to run your kinetic data on channel number, or you're only going to run your kinetics on your corrected data. But when you're doing kind of your checks and balances, determining how good of a scientist you actually are, it's really nice to look at all of the data itself. So before we go into specifics, the last one that I also like to load is your full test preview. So this is just the folder or the file that's underneath your 3 different resolutions. If you click on that and import it into trace drawer as well, we'll click OK. So the reason that I like to look at the full test preview is because if you look here, you can see everything that happened throughout the course of the test from start to finish. And it's presented for you in 3 different lines. The black line is channel number 1, so your reference. Red line is your active channel, and then your corrected data, which is a blue line. If you're doing a regeneration test, this is a really nice figure to look at to see how well does the baseline continuously go back to whatever level it was after your successive regeneration solution tests. So if you're doing a regen, I highly recommend taking a look at the full test preview first before you actually go into the individual injections. So the next thing I'd like to do, I've oriented myself. I have my full test preview. I'm then going to go to my channel 1 responses only. So you can see on the right-hand side, we have all of the injections that took place throughout the course of the entire test. And you have the option, you can select all of them. You can select individual ones if you'd like to highlight them or make them a little bit more bold. What I like to do is for my first check, I select them all and then right-click Hide Selective Curves. It removes everything. And then you can go through and you can look stepwise at each injection if you like, or you can focus in specifically on ones that are of interest to you. So in this case, I clicked on our ligand, which is the biotinylated oligo, and you can see it's a fairly flat line. By right-clicking and choosing Zoom to Extents, you automatically rescale everything, and this allows you to see how does my ligand immobilization look like. And if you're panicking right now, I hope you're not, because we're in channel 1 right now. We shouldn't have any ligand immobilization here. And you can see we're staying around the 0 mark. We've got a little bit of drift, but that's completely normal. So we're not having any ligand actually immobilized in channel 1. Next thing I like to do is go to channel number 2, which you just navigate to under the Add Run section. You would select all of your injections, right-click, hide them, and then look at your overall ligand immobilization. So I would just selected BT oligo, which is my ligand, Zoom to Extents, and you can see in this test we had about 175 RUs of ligand immobilization. A nice feature of trace drawer is you have the option to create as many figures as you like. So you click on the new overlay, and then you can go over here. You can rename it to be whatever it is you'd like to call it. You have options to change the scale as well. But what I like to use it for is to prove to my boss that I'm actually doing science by creating some very nice, quick comparative figures. So the first thing I like to do is show my boss, hey, look at this. I immobilized the ligand and it only went into channel number 2. So by doing that, you click on New Overlay, give it whatever name you like, navigate under the Add Run section. Right now, we're in channel 1, and I just have my ligand selected, so I dragged it up to the New Overlay. And then I'm going to go down to channel number 2, same thing, it's only the ligand selected, drag it up, and it's just going to superimpose them on each other. This is really nice because it's a really quick figure to show you, yep, I absolutely have no response in channel number 1. You do have the option to modify a couple of things. You can change the colors if you like. I like to sort it by run. So the red line is always going to be your active or channel number 2. Your black line is always going to be your referenced or channel number 1. And it just shows you, it's a really quick visual to say, great, I got it. I know I didn't immobilize my ligand in channel number 1. Another thing that I like to do is an NSB comparison. So I click on New Overlay. I would just change this. We'll call it NSB. And then what we do is we go to channel number 1. Instead of having your ligand selected, you're going to select the various concentrations of analyte that you did. You can zoom to extents and drag that up to NSB. And then you do the same for channel number 2. So 1, 2, 3, and drag this up. So when you're looking at it here, you can see we have 3 very nice concentration dependent responses, which is channel number 2, and then 3 that are hovering around 0, channel number 1. Once again, if you find it easier to see the colors visually, of course you see the colors visually. If you find it very easy to kind of compare things, you can change the colors depending on what channel they were inserted into. So here we have kind of definitive proof that there was no non-specific binding that was occurring that may obscure some of our kinetic calculations. The next thing that we like to do is to run your actual kinetic information or calculate your kinetics. So to do that, I caution you, you're only going to use the corrected version to do your kinetic calculations. The corrected version already has any responses from channel 1 subtracted out of the responses that were seen in channel 2. This is just going to standardize everything and help you derive more accurate kinetics. So from here, we're going to click on New Overlay. We'll call this whatever we want. We'll call it kinetics. Apparently, I'm not going to spell that right today. And then what we're going to do, navigate to corrected version. You can remove either all of them and then select just your analytes, or if you prefer to do it individually, you could just select certain ones. So we have only our 3 analyte concentrations selected. Drag it up into your New Overlay, and then you have a couple of options. You can right-click and choose Kinetic Evaluation, or you can click on New Evaluation here, make sure Kinetic Evaluation is selected from the drop-down menu, and then choose which overlay you would like to run your kinetics on. So in this case, we're using the Kinetic Overlay, and we're going to click OK. So what it's going to do now depends on what version of the software you have. I have an older version. It gives us a little bit more to do before it will generate the kinetics. If you're using the newer version, then the time and concentration changes are automatically put in based on the flow rates you selected in the OpenSPR. But for this, we're going to just go through it manually. So you can see along the bottom, we have a graph or a table, which is showing us the 3 different concentrations of our analyte, and they start at 0. This is normal because the software understands before the injection started, there was no concentration of analyte passing across. What we now want to do is drag this concentration change bar to where the injections actually began. So to do that, you just move the concentration change bar. You can draw a kind of square around a certain area of your figure in order to zoom in. And we're going to change the concentration bar to be where we first start to see responses in the data itself. You can do this individually. So for instance, if I just wanted to assign a concentration change for the 200 nanomolar, then right now only the orange line is being selected. Or you can do it for all 3 if they all are happening around the same time. So once you've dragged the concentration change to the appropriate place, you click Add Change on the bottom here, and you can see that the concentrations of your analyte are automatically put in. These are taken from your SPR, your OpenSPR software, where it's asking you what is the concentration of the analyte that you're injecting. If you made a mistake when you were building that, you can always double click and change this here. Or if you forgot, maybe you didn't put in a concentration, you're doing all of your injections, and sometimes it can become very, did I do that already? And you missed one, totally fine. You can put it in here. But what I like to do is just make sure that each of these is accurate. I was supporting a customer a while ago, and she unfortunately didn't realize that she had put in the wrong concentrations and her kinetics were way off. And it took us a minute to figure it out because everything looks so good data-wise. So you just want to make sure that everything here is accurate to the concentrations that you're testing. And then the last step to do is to drag your concentration change one more time to the end of the association. Some people will do this by math. If they know what flow rate they chose, they will just add that contact time to where they started their injection, and they know that that's where the injection ended. Other people will do it visually, depending on where the peak is and the dissociation starts. Or you can do it intelligently and upgrade your software, and it will do it for you. So clearly, I'm not in the intelligent group yet, but I will be very shortly after this webinar. So what we are doing here, we're just going to drag this over, set it to where we think the association ends, click on Add Change, and it reverts back to 0 because during the dissociation, there's no new analyte being introduced. From that, we click on Next here, and it goes to ask you what model would you like to choose. So we recommend sticking with a 1-to-1 model unless you know that your ligand is interacting with your analyte in a different way. When you're trying to orient what model am I going to choose, it's always from the ligand's point of view. So if the ligand is only binding one analyte, it's 1-to-1. If the ligand is binding 2 analytes, it's 1-to-2. So just think of it from the ligand's perspective. And once you've selected your fit model, you click on Settings, and there's 4 different options or 4 different parameters that you can use. So the Ka and Kd, what these are your on and off rates. They're set to global, and they should stay as global. The third option is your Bmax. So this is the maximum signal that you can see if all of your ligand-bound analyte. If you are using a successful regeneration, then you are going to change this to be global. So it's telling the software that after every analyte injection, there was the same amount of ligand available. So technically, we should be able to reach the same maximum signal. If you're not using a regeneration, then you would leave it as local. And that is telling the software that after each injection, there was less ligand available to be bound for my subsequent injections. The fourth parameter is your BI. So this is your bulk index. We set this to constant and then change the constant to 0. So the reason that we do this is because the bulk index, so the bulk shifts that we were talking about previously, if you're using a 2-channel system, they are automatically subtracted out from the corrected data because a bulk shift would happen in both channel 1 and channel 2. If you're using the corrected data, that's been removed. So by setting it to constant and that constant to 0, we're telling the software, don't worry about trying to fit any potential bulk shifts. So once you've set those, you're going to click OK. And then finally, you're going to click on the fit option right here. TraceDraw is going to automatically start the fit. Sometimes this can take 2 or 3 minutes. Sometimes it can take 15 or 20 minutes. If you find that it's taking a longer time, there's the potential that you didn't select the correct fit model. And so by going back and trying the fit with using a different model, it may increase the speed with which it's calculated, as well as increase the overall overlaying of the fit versus the raw data. But in our case, our kinetics have already been calculated. And what we can see here, so this new table is showing us our maximum signal, which is about 275 RUs. It's giving us our Ka, so K on, Kd, K off, and our affinity, so 4,4. And then further on, it's showing us our chi-squared signal. So the chi-squared is just kind of a goodness of fit measurement. We consider a nice chi-squared to be 10% or less of your Bmax. So if that's kind of in line with what you're seeing, then your fit is fairly nice. One other thing I like to do is click on Show Errors here, and it will introduce all of the errors for all of the parameters that you calculated. What we like to see is a error that is at least one order of magnitude less than the actual value you're working with. So for instance, here we've got e to the fifth, and the error is e to the 1. So these are what we would consider nice. There's -4 versus -7, or -9 versus -12. If you have large errors, it could mean that maybe one of the analyte responses wasn't in line with the other ones. Maybe the raw data itself was a little bit off. Maybe you didn't select the appropriate start and end times for your association and dissociation. There's kind of a variety of things that you can play with. We do have a support article on this as well. So if you're kind of saying, how am I going to improve my fit? You can always reference that or give us an email, and we'll give you some ideas as well. But what I like to look at now is the 3. So you can see we have the 3 raw data traces, and then the fits for each of them overlaid as well. So this is really nice. You can quickly visualize how well does my raw data fit with my actual model that I selected. And then you can either look at just the fits themselves, or just the raw data. So, but basically what we're looking at here, it took it away from me. It's fine. We'll just process it again. Is we want to see a fit that's fairly well overlaid with the raw data. And we want to make sure that the raw data itself has errors that are less than the calculated values, 1 order of magnitude or more, less than the calculated values that we're presenting. From this data, we would consider this one replicate. So the replicate itself, you can see that we have 3 different concentrations with a different fit, and we would report this KD for them. However, we then ask that you would repeat this test 2 or 3 more times to generate with using the same parameters, same analyte concentrations, generate KDs using independent fits, and then you'd be able to kind of build your own overall K on, K off, and affinity using multiple replicates of a test. All right, so we're not going to save that. When you're done, you do have the option, you would save it. It's going to automatically save under the same data file that your raw results or your raw data was generated in, unless you indicate that you'd like to save it somewhere else. So that is kind of the generalized overview of everything. The things that I'd like to leave you with is the support portal that I've been harping on about for the last hour and a half. I really do encourage you to check it out. It is free to register. So if you haven't registered yet, all you have to do is just put in your name, your email address, and what instrument that you have. So depending on if you have Rev 4, which is your manual instrument, or you can have a Rev 4 with your XT if you have the auto sampler. So just make sure that you're selecting that. And then the knowledge portal articles will be tailored to your specific instrument. You won't have to sort through the others. And the other one is, it's literally my job, it's literally Daryl's job to help you design, analyze, run tests. So if you're unsure, maybe you're seeing, okay, I'm not sure, will this work, or will this, is there something that could go wrong that I'm not foreseeing? You can always fire us off an email. We're happy to either email you back or to continue, maybe jump on a remote call real quick. And then we can kind of give you some input that we've had. And to contact us, you would just use the support@nicoyalife.com email address. It's sent to a general email that me, Daryl, and the rest of our team have access to, and one of us can get back to you very quickly. So that's, I really do encourage you to use both of those resources because we're happy to help you before and potentially help save a little time when you're trying to optimize your tests. So that is the end of my spiel. Look at that. I have 3 minutes to spare. I was so worried because I usually go over. I talk a lot. So that gives us some nice time to potentially look at any Q&A. I see that Daryl has been on top of everything.
Yeah, thanks, Rob.
Yeah, no problem.
Yeah, that was really great. Really great presentation. Yeah, as Rob mentioned, yeah, there's some time for Q&A. You can either type your questions and I can read them out. I've already answered a whole bunch of questions. Or you can also raise your hand and we'll allow you to, we'll sort of open it up for you to speak, and you can ask your question live if you'd like as well.
If there's no questions, I'm going to assume I did a perfect job. So I know that's not the case. So you might want to ask something.
Yeah, there's a question here. Thanks for the lecture. Here are some silly questions. They have a first generation SPR instrument, and typically they use 3 different concentrations of analyte. And is the final KD obtained by averaging the results of those 3 runs? This is to say you use 3 different concentrations. Do you would fit all those 3 together, then I'm assuming that's correct. I think if I'm understanding the question correctly, if you have 3 different, let me just say, if you have 3 different concentrations of the analyte, then you would fit all 3 of those together, just as Rob did, to get 1 KD. If you run the experiment 3 different times, as Rob suggested, then you would average those 3 different KDs to get your final KD, if that's answering it correctly. And then the second question is, does the first generation SPR, this is, I'm assuming this is the 1-channel SPR. Does that so Rob can answer this. Does the first generation or 1-channel SPR have a ligand wizard? They've never heard of this before.
Yes, it doesn't. So I apologize for getting your hopes up, but you can still use, there's a couple of support articles out there for what we call it Rev 3. That's what we refer to as a first generation SPR. So when you're on our support portal, the Rev 3 articles will give you an idea of how to tackle like testing for non-specific binding, or looking for will I be able to immobilize? You're not going to have the 2 different channels, so you're going to do your ligand immobilization in the channel after you do your non-specific binding test.
Yeah, that was her second question as well. What are the main drawbacks, and or third question, what are the drawbacks and disadvantages of a 1-channel instrument? So I think, yeah, you answered that as well. It's not having the reference channel there.
Yes. Yeah. And there's definite workarounds. It's nice. You can still use that channel to conduct a non-specific binding test, or to look for these other kind of experimental artifacts. It just will involve running a different series of tests before you start testing your main ligand and analyte pair.
Perfect. Perfect. Casey said, yeah, thank you. Okay, the next one is, could you explain how to interpret data for a competition assay? For instance, we're looking to see how a small molecule disrupts binding of 2 known binding partners.
Nice. Okay, so the competition assay, you have a couple of different options in how you run it. There's solution competition and there's surface competition. So basically what that means is if you're doing a solution competition, you're going to have, we'll call them A, B, and C. So if you think that A and B interact with each other and prevent binding to C, then what you would do is you would immobilize C onto your sensor, and then you would test pure, just so C versus A only. You'd leave B out of it, and you would generate kinetics for C versus A. Then in a subsequent test, you would have a standard concentration of B, and you would incubate that with increasing concentrations of A. And you would pass this mixture across immobilized C, and you would see, is there a difference in signal as A increases versus the standard? Does it basically overcome the ability of B to prevent it binding to C? And so once you have both the kinetics from your A versus C, so the pure kinetics, or your kinetics from C versus your mixture, you'd be able to compare those and see, is there actually an effect of my potential competitor preventing the binding between my 2 partners?
Yeah, perfect.
There's also, I know you guys are going to hate me. You're like, yeah, we know, Rob. But if you go on our blog, there's a great competition assay note, and it walks you through exactly what to do. So that's if you go on nicoyalife.com and then go on the blog and just type in competition assay, it will bring that up for you. And it will go over everything that I just said and kind of walk you through how you would determine which one you're going to immobilize and which one you'd like to test as the competitor.
Perfect. Any other questions? All right. Don't feel shy. Oh, wait a second. Yep, perfect. They're saying thank you.
Awesome.
All right. We'll give 10 more seconds for any other questions, but otherwise, thanks everybody for coming. This has been really great. I really enjoyed the presentation, and we'll have a video of this available if you'd like to watch parts of it again. And yeah, thanks everybody for attending. It's really, really great. Perfect. Have a great day, everybody.
Thanks so much, everyone.